this rapidly expanding field. All right. Okay, but first, we're going to go through a story. So I'd like everyone to meet Eero Mantai-Ranta. He's a Finnish Olympic athlete that was born in 1937. He grew up in poverty in rural Finland, and by poverty, I mean that his family of 6 lived in a tiny 170 square foot home together, and they would share a single fork that was passed around the table at mealtimes. He started to ski at just the age of 3, and he actually had to ski to school to and from across this frozen lake. And on nothing more than wooden planks that were nailed together. This trip was about an hour, and as you can imagine, in the winter, it would be pitch black, both ways. So the kids would, so from what I read, I read about this story in The Sports Gene, a book that was written by David Epstein. It's a really great book if you're interested in this topic. And one of the things that was mentioned was how, because it was so dark, the kids would pretty much just kind of aim for the opposite shore and just hope for the best. So in Eero's town, everyone pretty much had to ski because it was a mode of transportation. And while it may have started out that way for him, as a young adult, he took it up more seriously as a way to improve his life station. He ultimately landed a job as a border patrolman, which was great because this basically allowed him to escape the danger and drudgery of forest work. It didn't take him long to stand out. He was winning races as early as 7 years old. And then at 22 years old, he made the Finnish Olympic team. He went on to become one of the most successful Finnish cross-country skiers. He participated in 4 Winter Olympics spanning the 1960s to the early 1970s. As you can see, took home many medals, 7 Olympic medals, 5 World Championship medals. And one of the things I want to highlight about his performance, because this is very relevant to our talk, is that in the 1964 Winter Olympics, he won the 15-kilometer cross-country race by a whopping 40 seconds, which is a margin of victory that really has never been equaled. And the next 5 finishers were within 20 seconds of each other. So this is quite remarkable. And then that same year, he won the 30-kilometer race by a full minute. So, the question is, what made this legendary Finnish cross-country skier such a success? Well, during a routine medical exam as a teenager, a blood test showed extraordinarily high levels of hemoglobin and red blood cell counts. And as we know, normal male hemoglobin levels range from 14 to 16 grams per deciliter. Eero was consistently over 20 and as high as 23 grams per deciliter. His red blood cell count was 25 to 50% higher than that of an average human. So this combination gave him an increased oxygen carrying capacity, which, as you can imagine, would be a large advantage in an endurance type sport. So because he was actually healthy, there was little concern about these elevated levels. And over the years, these findings were actually repeatedly noticed or noted on his repeat medical exams. Now, like I said, he went on to compete in the Olympics, and that was mostly in the 1960s through the early 1970s. Blood doping didn't really start until the late 1960s, and it wasn't outlawed, actually, until the mid-1980s. So sadly, despite the fact that his blood levels had been documented since he was a kid, there was significant speculation ultimately, that his unusual blood profile was the result of doping. And then it wasn't until 20 years after he retired, so he had to deal with all of that. And it wasn't until 20 years after he retired from skiing that scientists were able to pinpoint the truth. Eero had a condition called primary familial and congenital polycythemia, basically natural blood doping. This condition was first described in 1933, but then we really didn't understand the underlying genetics until the human genome was sequenced. So this kind of came about more in the 1990s. And the cause of this condition is a rare gene mutation in the erythropoietin receptor gene on chromosome 19. It's an autosomal dominant single nucleotide polymorphism, in other words, SNP mutation. And just as a quick physiology reminder, if you look at the bottom of the screen on the right side, I wanted to just remind us that erythropoietin is a hormone that's primarily secreted from the kidney, a little bit from the liver. And this goes to bind to erythropoietin receptors on red blood cell progenitor cells in our bone marrow, and that stimulates red blood cell formation. So while this mutation was just a SNP, minuscule, the impact was immense, and that's because it created an early stop codon, which truncated the protein by about 15%. And remarkably, this caused the receptor to become hypersensitive to the hormone erythropoietin, which therefore increased the rate of erythropoiesis. And all in all, this is fortunately benign. It's not like the other primary polycythemias that you may remember from med school, like polycythemia vera. Those are myeloproliferative, and so they can dangerously affect blood viscosity and lead to strokes and thrombosis. And there you have it. You have a mutation beneficial for an athlete and otherwise of little consequence. Obviously, Eero had rare talent, and by no means do I share this story to detract from an athlete's hard work. Carrying this or any advantageous genetic mutation doesn't change the need to train incredibly well to alchemize that talent into Olympic gold. And in fact, other athletes have since been found to have that same condition. You can earn a medical exemption to compete if you're able to prove that those levels are natural. And actually, many have gone on to be very successful. So yeah. Let's fast forward to the early 2000s now, when the era of sports genomics began shortly after the completion of the Human Genome Project and sequencing of human DNA. So what is this field? It's relatively new. I would say that it's in its infancy, and it's focused on understanding the genetic architecture and biomarkers underlying the various aspects of athletic performance. So what's a biomarker? A biomarker is a biological feature or characteristic that's objectively measured and evaluated as an indication of either a normal or pathological process, biological process. They can be characterized in different ways, and genetic variations are just one of many types of biomarkers that one could potentially study in an athlete. So, for example, there are biometric biomarkers such as height, weight, BMI, molecular biomarkers such as ferritin and cytokine levels, vitamin D, and then from an imaging perspective, the bone mineral density can be an important biomarker. For genetic biomarkers, we're mostly focused on SNPs because this is the most common type of genetic variant among us. And so, this classification is based on a characteristic, but another way that we can classify them is based on application. And so, for example, these biomarkers could be relevant for diagnostic purposes versus screening purposes, prognostic or predictive purposes. So in other words, it's really the circumstance under which they're being used. While sports genomics wasn't born until the early 2000s, for decades, people were asking, are champions, or, in other words, elite athletes, are they built or born? And of course, dedicated training has long been known to improve athletic performance. But why do some individuals consistently outperform others who've undertaken comparable or even exact training regimens? And this question was often framed as the age-old nature versus nurture debate. So, again, are we born with the genes that will make us excel at certain sports? Or is it just dedicated training, a good diet, other types of lifestyle and environmental factors that determine success? And the suspicion that genetics played a role, it actually came originally from just pure observation. I think we can all relate to this when we think back to when we were younger in school. Maybe we had a friend or a classmate that was just a natural, that was just gifted, seemed to beat everybody every time with seemingly little effort. And then there were also the past trends of Olympic winners. And this was eye-opening, to be honest. The most notable of these phenomena is the African runners phenomena. This intriguing phenomena is really what sparked interest in that question and debate, with regards to this question on a global level, right? So athletes of African descent were noted to sweep all the running events and essentially hold every major male running record from the 100 meters to the marathon. And that same trend actually holds for female runners as well. It's just not as pronounced. It's not as dominant as it is with the male runners. Meanwhile, Caucasians and Asians were dominating in water sports, field competitions, strength events. And if you zoom in on this phenomena closer, you can see this remarkable mirror image success story. So over in East Africa, we have the champions of endurance, where Kenyans and Ethiopians are dominating in the middle and long distance running events. And then on the opposite side of the country in West Africa, we have the fastest humans on Earth. Of course, we're all familiar with Usain Bolt, also known as Lightning Bolt. He was a Jamaican legend who to this day holds the world record in the 100-meter race, coming in at 9.58 seconds. And you may wonder, well, he's Jamaican. And when I look this up, as it turns out, most of the ancestry for Afro-Jamaicans actually comes from West Africa. So the other thing that one might kind of to play devil's advocate, one might say, well, East Africans live at higher altitudes, so could that maybe explain this phenomena? And honestly, maybe, but the thought is that this trend is just so stark that it's unlikely to alone explain that. And then up until recently, and I mean like 2020 recently, there were no elite level Asian sprinters, none. And intriguingly also, there were none from East Africa. However, everyone has eyes on Su Bingtian, and I apologize if I mispronounce that, but he is Asia's fastest sprinter ever. He's the only sprinter of non-African descent to run the 100 meters with sub 990 and 985 second times. He's the first Asian-born sprinter to break that 10-second barrier for the 100 meters. And in 2020, at the Summer Olympics in the 100-meter final, he made history becoming the first sprinter of non-African descent to enter the 100-meter finals since 1980. So it'll be very exciting to see how he does in the Paris Olympics in 2024. But back to our question about the suspicion of whether genetics plays a role in achieving that elite champion level athleticism. So to summarize, we talked about our personal observations when we were younger and growing up. There's the African runners phenomena. And then of course, let's not forget Eero's story that I started off with. So we come back to this question, are champions built or born? And as it turns out, the answer it's not such a black and white question, basically. Thanks to advances in our understanding of genetics and human traits, this really isn't a debate anymore. And we know that it's actually a little bit of both. So in other words, the consensus is that many external factors affect athletic performance, like training, diet, skill development, climate. And the confluence of those external factors may shape athletic development, of course, but the key driver to being a champion is thought to be intrinsic. So in other words, those that are at the very top have a higher limit based on their genetic potential. And so the hunt for sports genes begins. And this has proven to be very challenging. Before getting into this hunt, I wanted to review some basic genetic concepts, which will help highlight why this has been so challenging. So the Human Genome Project took about 13 years, and it was finally published and completed in 2003. Our genome, our diploid genome, consists of 6 billion base pairs. We've got 25 to about 30,000 protein coding genes. And what's crazy is this only makes up less than 2% of our entire genome sequence. Our genome is a double helix of nucleotide base pairs, so adenine, thymine, guanine, cytosine, and it's wrapped around histone proteins like a spool of thread. And this is what forms chromatin. And then that chromatin is further compressed into 23 chromosome pairs, including 1 pair of sex chromosomes. Our mitochondria also have their own small genome. So when we think about how different are we really, our DNA is actually 99.9% similar to one another. So where do we get all this variation? And well, about 0.1% difference, which you might think is very small, it actually equates millions of base pairs. And thus there's a large number of potential unique sequences that can come from that. But then where this potential for variation gets taken to a breathtaking level is when you factor in something called epigenetics. Epigenetics is basically the study of changes in gene expression, so the phenotype, that don't involve changes in the actual underlying DNA sequence. And our DNA sequence is known as the genotype. And this is done through a variety of different ways. I mean, the regulatory complexity of our DNA is immense, but it's done through things like DNA methylation, histone acetylation, non-coding microRNAs. I'm not going to get into the details of all those things in my talk, but I just wanted to highlight like the complexity of it all. And there's gene-gene environments, gene-gene interactions and gene environment interactions, which I'll touch on in the next few slides. But epigenetics is reversible. So unlike our DNA sequence, our genetics, which is not reversible, the sequence is there and it's fixed. This is reversible, and lifestyle and environmental factors will modify epigenetic patterns. So things that we eat, our diet, our physical activity, smoking and alcohol consumption, toxins that we may be exposed to, psychological stress, and even the uterine environment has been found to be important with regards to traits that the baby ends up having. And so as we've come to understand all of this, we now realize that, again, our DNA sequence may be fixed as a sequence, but the code isn't. And so the choices that we make and the things we're exposed to can affect the phenotype. And an analogy that I like to give is if you think, for example, about a standard piano keyboard, which has 88 keys, it's fixed, that's all there is, there isn't going to be any more. And epigenetics is essentially the piano player. So it's someone who's able to sit down at that same keyboard and play jazz music, classical music. It's just a different, different songs, different types of music. Human traits are typically, or generally we think of them as either Mendelian versus complex. Mendelian is monogenes, so it's a single gene that's involved, and it follows a Mendelian inheritance pattern. In other words, autosomal dominant or recessive, or it could be sex-linked. And usually these types of traits are not influenced by the environment. So for example, some of these are like our blood type, freckles, people who have we have either free or attached earlobes, a cleft chin or dimples. These are all essentially single genes not affected by the environment. And then on the flip side, we've got polygenic traits, which involve more than one gene. And these are usually influenced by the environment. And this is the majority, to be honest with you, of things when you think about human traits. It's our height, skin and eye color, the ability to roll your tongue or not. And most human diseases sit in this category. Abstract traits like personality and intelligence, and then last but not least, athletic performance. If we think about, okay, let's dissect the Olympian. What does it take to get there? Well, we know that, okay, there's a component of genetics, and we know that that genetics is both our sequence, but also the epigenetic component. So there are gene-gene and gene-environment interactions that are happening, which again, I'll get into in the next slide or two. But then there are also pure environmental influences. And the way to think about that is, well, to win, it's going to take a certain amount of motivation, dedication, perseverance. What's the drive? It might be kind of depending on where you are in life and your family. The need to become an Olympian is different from one person to another. So someone may be way more driven to go down this path than someone else, right? The quality and the hours of training, the equipment that they can use. So all those things are just like pure environmental influences, genetics aside. To give an example of the gene-gene interactions, this is essentially where you have one gene that codes for a protein, and then that protein goes on to affect the transcription or expression of another gene. And an example would be, for example, a gene that causes albinism. This can hide the gene that controls hair color. Another example would be a gene for the widow's peak and how that would be hidden by genes that drive baldness, for example. And then an example of a gene-environment interaction is this interesting phenomenon that we see in Siamese cats. They have a temperature-sensitive mutation, and that mutation is in the TYR gene, which encodes tyrosinase, an enzyme that's located in melanocytes, and it's responsible for melanin production. And for reasons that we don't clearly understand, this mutation renders the protein less stable at higher temperatures. So if you look at the Siamese cat, you notice that the peripheries are darker. There's more melanin, the ears, the nose, the periphery of the limbs and the tail, but then down by the torso and the belly, it's lighter in color. Another reason athletic performance is complex is because it's comprised of a multitude of parameters, right? It's so vague and so broad to say athletic performance. Things like strength and speed and power and balance and coordination, agility, there's so many different parameters. And each of these parameters have their own set of genes that influence them. And then collectively, they're all contributing to athletic performance. So again, kind of makes it tough to study this. But the technical approaches generally to genetic research are genotype-phenotype correlation studies. So essentially what you're doing is you're aiming to test whether a given genetic variation, for example, SNP, and whether that's involved in controlling the phenotype of a specific trait. So, for example, athletic performance. The two types of studies that have mostly been used have been candidate gene association studies and genome-wide association studies, which I'll kind of refer to in the future as GWAS studies. So the candidate gene association studies were the first type of studies that were done in the realm of sports genomics, and they actually still make up the vast majority of the studies that are out there in this field. They're hypothesis-driven case control studies. So again, you're thinking about, well, I want to study athletic performance. We know that tendons and muscles are important. So let's go target genes that we know are involved in the tendon apparatus or muscle fiber type. So we'll go study those, look at specific allele changes between cases and controls, athletes and sedentary people. These studies are inexpensive. They were relatively easy to do. However, they fell out of favor fairly quickly because, there were several limitations. You could only look at one or a few genes at a time. The cohorts were small. They were about 10 to a few hundred. or tens to a few hundred. And for that reason, they were underpowered and there was a high false positive rate. So the other key limitation to them is that they only focused on coding DNA, right? Because it's this a priori hypothesis where you're thinking, okay, I'm going to focus on tendon genes. So you're already just, excluding the vast majority of our DNA sequence and that vast complexity of gene regulation that I was referring to earlier. To give you an example of some of the genes that were being studied, again, you can see they focused on things that looked at endurance capacity, muscle performance, the tendon apparatus, and even genes that are thought to be involved with psychological aptitude. I'm deferring going through any of the data because, like I said, there were a lot of high false, it was a high false positive rate. So we just don't know if it's accurate. Now, genome-wide association studies. So candidate genes were the a priori hypothesis-driven efforts to pinpoint a specific genetic pathway. GWAS studies instead, they start with the more general premise that genetic factors are important to athletic performance, our trait of interest, but we don't know exactly where to look in the genome. And so we're going to look broadly, almost randomly to find places that are important. And this is the more modern and now main method of determining genotype-phenotype associations. Because they use microarray and high throughput sequencing technology, we can now genotype millions of pre-selected SNPs across the genome. And there have been a handful of these studies so far. Again, the data, this is all still a field in its infancy, so the data remains preliminary. But the advantages that this type of research approach offers is that the cohorts are much larger now, so thousands. You're also scanning the entire genome. You don't have to just focus on one or two genes. You're also not just limiting yourself to the protein coding regions of our DNA, of our genome. And for that reason, it's afforded a considerable advance in the power of the analyses. One important offshoot of sports genomics research has been to examine the susceptibility of sports-related injury, and most of the studies were focused on soft tissue injuries like tendons and ligaments. And I was fortunate to be a part of a group of researchers that was doing some of these earliest studies. Most of our studies found like maybe 1 to 2 SNPs that were of genome-wide significance, but this needs to be replicated. So again, I would say that this is still preliminary and we can't form any definitive conclusions, but you could see we looked at MCL injuries, Achilles, ACL, rotator cuff issues. Thanks to the tremendous advances that we've had in DNA sequencing technologies and the reduction of costs, there's this new direction of potentially just sequencing the entire genome, not just focusing on parts of the genome. And so that could be very exciting. And then consortia approaches are gaining steam. And so this is essentially bringing together brilliant minds, all the resources that they have, and combining this to form larger biobanks of data that could be studied. The Gen Athlete study, for example, is looking at endurance athletes and VO2 max. The Athlome Project Consortium is the most, is the newest consortium formed in 2015. And they're really hoping to form the largest biobank of athlete samples. And I think it's important that this is happening because, as the evidence has emerged that genetic influences are more likely to be many genes, each of which has a small impact, as opposed to what we saw with Eero Mantai-Ranta's story, where you had this one gene and large impact on performance. So, I think there was a lot of excitement when the human genome was first published, because they thought, wow, we're going to go out there, we're going to find gold medal genes, champion genes. It was going to be so easy. And what we're seeing now, like that kind of excitement has dissipated. It's much more complex, and we're really looking at the many genes, small impact variety, and that's going to require huge cohorts to be able to catch these statistically significant differences. And so that creates a problem, because one is we don't necessarily have that. And even if we had the resources to try to pull that together, elite athletes are rare. Where do you find thousands upon thousands of elite athletes of that particular sport? So it's likely going to require that we stretch the definition of elite and start combining like not just Olympic and level athletes, but regional, national athletes, and then also combining athletes from different sports that have similar underlying physiologic traits. So we could look at endurance runners with cyclists and cross-country skiers. I've already touched, of course, on some of these challenges that we're facing in determining genetic determinants of human athletic performance. But just to summarize, again, it's a polygenic complex trait. So we're looking at intrinsic factors such as genetics, but also extrinsic factors such as training and nutrition. And then there's the interplay, the epigenetics of all of that. There's also debate regarding the magnitude of genetic influence on sports performance and how training and diet can potentially overcome the influence of any advantageous or disadvantageous genetic SNPs that someone might have. Many traits are influenced, like I said, by an interplay of large numbers of DNA variations. So it's that many genes, each of a small impact. And again, we don't have thousands of athletes, elite athletes to study, elite runners. So the same physical outcome as well can sometimes be reached via many different genetic pathways. And then last but not least, to throw a wrench in all of this, as if it isn't complex enough, is the gut microbiome appears to have influences on performance. We know it has influences on disease, that's not new, but it also seems to have influences on athletic performance. And so we may need to start looking beyond our own human genome to fully understand our own potential, physical potential. What are some of the implications of sports genomics? Well, one is certainly it's important because it could help us improve our own knowledge of our human physical condition. And that's becoming all the more important as the human lifespan lengthens. And we all want to have just as long a good health span, right? We don't want to just live longer, but be really sick towards the end. It's also important when we think about detecting sports talent. And this may help identify individuals with a natural predisposition to achieving success in specific athletic activities. It may help us personalize training in a way that we can optimize performance, recovery, injury prevention in all levels of athletes, including the military. It may help shed light on underlying pathophysiologic mechanisms of musculoskeletal injury, which is important for prevention, but it may also help guide treatment approaches. And then in the identification of athletes at risk of specific types of injuries in pre-participation screening. So right now, the NCAA has mandated sickle cell testing, which is great. But if we can further our knowledge, we may be able to have actual genetic biomarkers that can help us with regards to concussion and hypertrophic cardiomyopathy. And then last but not least, it would be fun to just unravel determinants of sports excellence. There's been, over the past couple years, a floodgate of direct-to-consumer companies that are trying to get in on this. And the question is, are they getting ahead of themselves? Is genetic testing for athletic ability a real science right now? Do we have enough, or is this snake oil? And I'm going to actually say that this is kind of snake oil right now. In the age of personalized medicine, there's a rapid expansion, or there has been a rapid expansion in the number of direct-to-consumer companies that want to try and, again, provide people with actionable health and wellness information. I'm sure a lot of us are already familiar with 23andMe. AxGen is one of the more recent ones out there, but it's not too expensive, a few $100s. They'll send you a kit, you send them back some saliva, and they'll basically run that for a handful of markers relevant to sports injuries, nutrition, and performance, and kind of spit something out to you. And in my opinion, can you draw any major conclusions? Again, any actionable health and wellness information that can come out of this? I think it's way too premature. It's not supported by the current state of the literature. And then there's also some ethical issues that will come up. So a consensus statement was released in 2015 and published in the British Journal of Sports Medicine that essentially discouraged this. All right, and so getting into, as we kind of come to the conclusion of my talk here, I wanted to go over some cautions and considerations. There are concerns that discovering genes that influence athletic performance will detract from the hard work undertaken by athletes, and especially if those genes turn out to be more concentrated in one ethnic group or region than another. And then there's also gene editing and gene doping. So of course, we've got gene therapy now that's enabled the treatment of several rare genetic diseases. This certainly can make its way into sport, but it's likely decades away, far away. And certainly it would be banned. It has yet to be documented as something that's ever occurred. But in theory, it might allow athletes to increase endogenous production of proteins that enhance strength, endurance, muscle repair, and it may be a way to gain a competitive edge. I wanted to also kind of throw in this controversy of what does a level playing field really mean when you think about it? Because going back to my original story of Eero Mantai-Ranta, his blood was naturally doped, and that was acceptable because he was able to prove it, but it doesn't change the fact that he has a competitive edge. And then Lance Armstrong became a pariah when he exogenously doped his blood, right? So as the rule stands, having an incredibly rare gene mutation that boosts red blood cells is okay. Training at altitude to boost red blood cells is okay. Shelling out thousands of dollars to sleep in a tent that simulates altitude is okay. Although the World Anti-Doping Agency is starting to frown upon that, and who knows, it'll be interesting to see if they actually ban it in the next couple of years. But then injecting or kind of exogenously affecting your performance it's not okay. This is definitely not okay. You're banned and you're a disgrace. And Lance Armstrong was stripped of his medals, as we all know. So I'm not providing a stance on this. I'm just purely kind of shaking the foundation and getting us to think about, what does a level playing field really mean? I think it's a really tough question. So where do we draw the line, right? How do we define natural human effort in sports versus, what's a fair advantage? Where does that end and where does cheating begin? I'll go through some summary points here, which is basically that sports genomics is a rapidly growing field, still in its infancy, focused on identifying genetic determinants of athletic performance. And even though we've sequenced the human genome, much of it remains a mystery to us. Sports performance is an enormously complex, polygenic, multifactorial phenomena, makes it really difficult to study. But despite those challenges, the future of this field is really promising. And the research is really growing at a rapid rate. Instances in which a single gene has a dramatic effect, like Eero Mantai-Ranta's case, is incredibly rare. That's not how it's most likely going to pan out. We're going to be looking at the many genes, small effect variety. And then last but not least, I think it's important to acknowledge and recognize that the existence of talent and of genes that influence athletic potential, in no way detracts from the work that it takes for that talent to be transformed into achievement. And there are broad implications in this field, ranging from identifying sport talent to optimizing training, reducing injuries. And last but not least, with the direct-to-consumer genetic testing companies that are coming out, it's important for us to just be a bit cautious about those things and until we have more robust evidence. Thank you so much. These are my references.
Thank you very much, Dr. Ahmed. We can open up the floor for questions. That was fantastic.
Thanks.
People can unmute their microphone, or you can put something in the chat. I'll keep an eye.
I'm going to jump in since nobody's saying anything. Dr. Ahmed, that was really fascinating. And it almost brings you to the point of saying, should we be looking at athletic performance as an art rather than a competition in many ways? It's fabulous what people can. There's nothing better than watching a top level athlete because they're beautiful to watch. On the other hand, the competition is so detrimental on so many levels, physically, psychologically, culturally. I mean, there's so much devastation, I think, from athletic competition. I almost wonder about, culturally and ethically, if we should be looking at athletics in a different way.
Thank you so much for sharing that perspective. I agree. I really do. I think it's fun to want to understand human physiology and science and medicine. But you're right. We don't want to lose sight of just doing things for pure enjoyment. And going back to what I was on the slide where I talked about the implications in identifying sport talent, in some ways it's like, well, I kind of don't care if I was dealt genes to be a great runner. I like to swim, and I'm going to keep swimming. So I think also we don't want to necessarily be having that override people's inherent, choices.
Hey, Dr. Ahmed, this is Donaldson. I'm one of the sports faculty here. So yeah, thanks for that talk. It was really interesting, something I haven't delved into very much. But a couple of thoughts that, number one, kind of what you're just touching upon, like whether identifying good genes and whether it detracts from people from wanting to train that hard. I think it seems like a lot of this research leads to a lot of kind of ethical questions. So my question, for example, another one would be in the future, if you can alter genetics a little bit, when do you draw the line of when that's, cheating, kind of like anabolic steroids right now? So my question to you is while you're doing all this research, are there a lot of ethics discussions going on, and kind of how do you guys tackle that? Is there an ethics committee that you guys are always doing concurrently, or how do you tackle that portion?
Oh, thanks so much for that question. Yeah, so my role mostly has been consulting, especially for those GWAS studies that I was telling you about, where we were looking at any genetic factors that might determine a risk of a specific musculoskeletal injury. There's definitely, IRBs involved, but no, we didn't have an ethics committee.
I think it's one other just thought is, in general, if you guys can do this more in mass, it would be interesting to see how many people who aren't elite athletes have the same genetics, and how that ends up corresponding with because right now, since you guys are screening primarily elite athletes, you guys are finding these traits in people who are already elite athletes, but how many people out there that aren't elite athletes have these same genetics that just aren't using them, or they just get displayed differently?
Totally. Yeah, and it's fascinating to think about, also how much can your diet and your training and your environment override any kind of disadvantageous SNPs that you might have, right? there's gonna, like, where this, well, it's already there now. It's already there now where they use genetic risk score. So it's like a way to calculate, well, there's this, plus, and I'm sure that's how it will continue to carry out in the future.
We have a question in the chat from
Yeah.
Dr. Cabrera. This is asking, what about cultural and financial variables? In the US, there's virtually no money for Olympic sports. So most genetically, quote, gifted athletes go into football, baseball, basketball, et cetera.
I don't know. I'm not really sure. I honestly not sure I can answer that question. Yeah, just because I think that these consortia, for example, that are coming together, have their own funding, and yeah, I just don't know. Sorry.
Yeah, thank you so much for. I think that this lecture is very provoking kind of a lecture to us, but I am not good with any genetics. But I have been very fascinated by epigenetics. I barely understand, but I was curious if we are using this epigenetics to improve their quality or quantity of their skill sets. So that when you examine the athlete's blood or whatever genes, and then are you guys, any researcher are using this epigenetics to improve their skill sets or their ability?
There are a couple. For example, ferritin is a big one. Vitamin D. There have been like a couple of studies that I think like female soccer players, and I can't remember. I think it was ferritin. And vitamin D is a big one, and also omega-3, which, like through fish oil, for example. And that brings in a really yet another really interesting field called nutrigenomics, nutritional genomics. And yeah, so there've been a few, they're small studies.
The reason why I had this question is because, as you know, I am crazy about cerebral palsy, and they have a significant problem with their strengths, right? They're very weak. So I was thinking about what if we use this epigenetics to improve their strengths. So that's why I was curious. I don't know, you have any idea, any research that related to cerebral palsy with the epigenetics?
That's an amazing idea. No, I don't. I'm not aware of the literature with regards to cerebral palsy, or even with just muscle strengthening per se. I don't think there are any large, really believable studies on that. But at the end of the day, I think things like vitamin D, fish oil for omega-3 fatty acids, ferritin, and magnesium, for example, some of these are vitamins and minerals that we are nationally, epidemically deficient in. And so they're low risk, right? To supplement them, they're certainly not going to harm.
Thank you.
You're welcome.
We had another question in the chat box for you, Dr. Ahmed. Dr. Prakash, what could be the best sample size if we talk about GWA's studies for a particular athlete or game?
Oh, yeah. I mean, right now, you could, at the very least, it could be thousands, right? Our GWAS studies, for example, were generally, the biobank we were using was like a little over 100,000. So there's, in some ways, there isn't really a limit. I think that the limit really comes from just how many, it comes from the actual biobank.
But like if, yeah, so Dr. Ahmed, it was a really nice brainstorming talk, but I have a question, is for a particular game, like if we talk about the marathon runners or the like short distance runners. So then if we take about the like for the we conduct a Jiva study. So like in usually Jiva studies, we talk about the 1,000 or 2,000 or 5,000 samples, at least minimum. But if we do not have access to biobank, or in biobank, if we do not have DNA samples from athletes, so then, and if we want to conduct a prospective study, so what could be the sample size? Because it's very difficult to recruit 1,000 athletes for a particular trait, that they are long distance runners, short distance runners, or they are like swimmers, or any kind of game. And then recruit means, if we talk about to recruit controls, like who do not participate in those games, then it's very easy. 1,000 or 2,000, you can get it very easily. But if you talk about a like case, like if we take it as a case, so then it's very difficult, I think, to go for 1,000 samples. Any recommendations here?
I wish I had. I'm not a statistician, so
Okay.
I don't know the answer to that. I can just tell you that, like, for example, in the studies we did, where we were looking at a little over 100,000, at most of our studies, we picked up like 1 or 2 SNPs.
Okay.
I think if we look at even a larger cohort, we may pick up more, and then for that to be replicable and statistically powerful, again, like the more.
Right. But do you have any idea that there are some biobanks who have some samples from the athletes, and then there we can procure this, the DNA samples, and can perform the analysis? Is it feasible?
Wait, sorry, repeat the question a little
Like,
more.
yeah, so like if we take if we want to conduct a Jiva study, and then we take the example of a particular game or a particular athlete, so then are there any biobanks where we can get the samples for a particular, like this particular means people like who are playing a particular game, so that we can look for the muscle like SNPs related to that particular game or the trait?
Yeah.
So like, yeah, in the biobanks where we can get easily samples, because if we think of recruiting the in prospective study, like for 2 years, 5 years, it's very difficult to recruit 500 samples from a particular for a particular game. But are there any biobank recommendations, if you have any in mind?
You are absolutely correct. Yes, that would take forever. So yes, there are. Those are the consortia that I was telling you about. Yeah, like the Gen Athlete study, the Athlone project is the most recent one, and they're really merging a lot of those consortia. There's a little bit of overlap between them. They're kind of using similar biobanks, but not totally. But I don't have like exact names to give you. I can tell you the one that we research. Well, my GWAS studies with my colleagues, those were done based on a biobank that was put together by Kaiser Permanente, and it was not a biobank of athletes. It was just Kaiser patients in that system, because we were looking at genetic markers for specific injury, sports injury, so ACL tears, for example. But you're right, the direction is going to forming biobanks that are purely like elite endurance runners or elite sprinters and so forth.
All right, thank you.
They exist. I don't have the exact names for you though, but those consortia, if you look them up, they'll say.
Okay. No, I had just question for the like recruitment of the like large sample size, but yeah, this answered me, so we can think. Thank you.
There are probably banks, for instance, for some athletes with concussion, Ravi, but the problem is they have concussion. So they get the biospecimens, right, taken after they have a concussion. So but I think there's probably some biobanks with football players and concussion, and maybe there's some, that's probably.
Thank you, Dr. Bell.
You're welcome. Thank you.
Okay, thank you, Dr. Ahmed. I think we're almost out of time here. If there are any last minute questions anyone has, feel free to speak out now. It doesn't look like it. That was a really interesting and thought-provoking talk. I really appreciate your time this morning. Let me turn my video on here. I think everybody really enjoyed that. nobody else has any questions, we've got great response in the chat box. Terrific presentation. Thank you. That was great. Once again, thanks for joining us this morning. And no one else has any questions. We'll maybe take a
Thanks
little
for the
break
great
here.
questions.
Yeah. Thank you everyone for participating. Dr. Ahmed, thank you again. And we're going to take a little break here before our next lecture with Dr. Bell coming up. We'll be presenting on mild concussion or mild TBI rather, and concussions. If you want to take a little bathroom break, get up, stretch your legs, get a glass of water. Dr. Ahmed, thanks.
Thank you. Thank you so much, everyone, for the interest and great questions. Thank you for the invitation.
You're very welcome.
Right.
All right,
Bye.
take care.
Bye.